Polysiphonia occurrence. Most of the species are lithophytes, i.e., found growing on rocks. Some species are epiphytic, found growing on other plants and algae. Some species are semi-parasitic. Thallus structure. The thallus is filamentous, red or purple-red in color. The thallus is multi-axial and all cells are connected by pit connections, hence, the name given is Polysiphonia. Due to continuous branching and re-branching, the thallus has feathery appearance. The thalli may reach the length of about 30 centimeters. The thallus is heterotrichous and is differentiated into a basal prostrate system and erect aerial system. The prostrate system creeps over the substratum. Its functions are attachment of the thallus to the substratum. Cell structure. The cell wall is differentiated into outer pectic and inner cellulosic layer. The cell contains a large central vacuole which is delimited by a membrane tonoplast. The cytoplasm is present between the cell wall and the central vacuole. The cell contains a number of red discoid chromatophores which lack pyrenoids. The chromatophores contain pigments chlorophyll A, chlorophyll D, carotene, R for chorethrin and R ficocyanin. The cytoplasm contains granules of fluoridine starch as food reserve. Reproduction. Polysiphonia is mainly heterochoric. In life cycle of Polysiphonia, both asexual and sexual reproduction takes place. The life cycle shows alternation of generation. Sexual reproduction. Sexual reproduction is oogamous type and plants are dioecious, i.e., male and female sex organs are produced on different male and female gametophytic plants. Male gametophyte. The male sex organs, spermatangia or antheridia, develop on fertile trichoblasts present on tips of male gametophytic plant. Development. The cells of fertile uniaxial trichoblast divide to form pericentral cells. The pericentral cells form spermatangial mother cells on outer side. Each spermatangial mother cell cuts off 2 to 4 sporangia on outer side. The complete structure makes cone-shaped cluster of spermatangia. The mature spermatangium is a globular or oblong, unicellular structure. The uninucleate protoplast of spermatagnium forms a male gamete or spermatium. The spermatium is non-motile and is released through an apical pore in the spermatangium. Female gametophyte. The female sex organ of Polysiphonia is called as carpegonium. The carpegonium develops on trichoblast on female gametophytic plant. Development. The trichoblast initial arises from a cell, 2 to 4 cells behind the apical cell. It develops into 5 to 7-celled female trichoblast. The 3 lower cells form 5 pericentral cells, of which there is 1 adaxial, 2 lateral and 2 abaxial cells. These cells surround the central cell. The adaxial cell, called supporting cell, forms a basal sterile filament initial, a lateral sterile filament initial and a curved 4-celled carpegonial branch. The basal swollen flask-shaped cell of the carpegonial branch functions as carpegonium or egg cell, and the upper tubular elongated part is called trichogyne. The sterile sheath around carpegonium is called pericarp. Fertilization. The spermatia are carried to the trichogyne of carpegonium through water currents. The spermatium adheres to the trichogyne by the mucilage around it. The walls between spermatium and the trichogyne dissolve. The male protoplasm enters carpegonium through trichogyne. After fertilization of male and female nuclei, a diploid zygote cell is formed.